We interpreted the results of a specimen stain to list the most likely organisms present in the specimen. We described the basic process of microbiology testing for identification and susceptibility. Now, let's identify the normal human flora and their role in infectious diseases. Now, normal flora are absolutely essential for health. There are non-sterile sites in the human body where these flora reside. However, there are sterile sites that are not supposed to have any organisms in healthy hosts. So in general, all internal organs are considered sterile. So, they should not have any flora. For example, in blood, we don't anticipate to have any organism. So if you do find an organism in this sterile site, they're considered in infection for the most part. So, especially blood, if you find anything in the blood, it's considered infectious. The same with the central nervous system, cerebral spinal fluid, in the liver, spleen, kidneys, lower bronchi and alveoli, and the bladder for the most part. So the bladder is a bit tricky because, especially in young men, the bladder is supposed to be sterile. But as part of aging, for most people, as we get older, we start to have colonization of bladder. That may, as people get older, bladder is no longer sterile. But, a young man, it will definitely should be sterile. In women, it's common for women, given the anatomical differences between men and women, that the bladder will be colonized, and women as they get older, also the percentage of colonization goes up. And anybody who has a urinary catheter almost certainly has colonization of bladder. So the bladder is kind of tricky on this list, but the rest of this list is pretty much, if you find an organism, it's likely due to infection. But let's focus on the normal flora in these other sites. So the skin, in general, has gram-positive aerobes, such as, any Staphylococci species, including Staph aureus. So, if you find Staph aureus on the skin, it's not necessarily infection, in a healthy host. Occasionally, this may cause skin infection. But in the absence of any signs and symptoms of an infection, it's okay to have these organisms on the skin. That's part of the flora. There are obviously oral flora in the mouth. So, Fusobacterium, Peptostreptococcus, and Viridans streptococci. And then for nasopharynx, again, Viridans streptococci, Staphylococcus aureus. And the reason I have bolded some of these are the ones that are clinically important, so you should definitely know them for sure. And this is important because, so let's say somebody has an infection and you find Staphylococcus aureus in the nose, it doesn't often mean that it's an infection, so it could be part of flora, but if you detect it, you can kind of anticipate, because not everybody will have it as part of their flora. So, it's good to screen patients just to see if as part of their flora they had Staphylococcus aureus. So if you have infection in other parts of the body, you can kind of anticipate that Staphylococcus aureus found its way to other sites to find an infection. So perhaps the lower part of the respiratory system causing pneumonia, and we will talk about those scenarios throughout the course. Let's see. So the anaerobes in the nasopharynx are the same anaerobes as the mouth, and there are some gram-negative aerobes in the respiratory tract. So H. flu, Haemophilus influenzae, and Neisseria meningitidis is one. And then in the vagina, there are flora. For example, E. coli is a resident of vaginal tissue, and group B streptococci and listeria. Of course, when people mostly talk about flora, they typically talk about the GI tract, so gastrointestinal tract, commonly referred to as gut flora, and there are a lot. So you can see on the right hand side that because of the harsh acidic environment of the stomach, not many bacteria can live in the stomach, and that's in general a defensive mechanism. And as you go down the GI tract, it's still acidic throughout, but the acidity decreases down, through the small intestine and then the large intestine. So you can see as the acidity decreases, the number and variety of different flora increases. So especially in the large intestine, you will have a diverse body of flora, and this is a good thing, right? So the more you have these healthy organisms in your gut, they prevent through competition other problematic organisms from growing. So you're less likely to have an infection in the GI tract. Most commonly what we worry about is Clostridioides difficile infection. So because these organisms will compete with C. diff, and it's healthy to have a healthy number of flora here. And this is also the reason why we are very careful when we choose antibiotics to treat other infections, because pretty much majority of antibiotics will affect the normal flora. So we want to avoid unnecessary antibiotics in order to preserve the flora. And when we cannot avoid antibiotics, we want to make sure that we use them for the shortest amount of time in order to minimize the collateral damage caused by antibiotics. But so you can pretty much see that these are mostly Enterococcus faecalis, which resides in the GI tract. Different species of Clostridium. A lot of these are anaerobes because this is an anaerobic environment for the most part. But some aerobes can survive here too. So Enterococcus is definitely aerobic and can have a big role here, as well as members of Enterobacterialis. So E. coli and Klebsiella certainly reside here. And the most important anaerobe is perhaps Bacteroides that resides in the lower GI tract, but pretty much unlikely to be found in other parts of the body. And in general, when people talk about antibiotics and ask if an antibiotic has anaerobic activity, they're primarily talking about Bacteroides rather than other types of anaerobes. Now, some important clinical concepts for interpreting cultures is, colonizer and contaminant. So anytime you have something growing in a culture, you need to ask yourself, is it coming from the source of the specimen, or did it get contaminated in the process when it was being collected? So a contaminant will not be from the site of the specimen. It could be due to improper technique, and then the colonizer is, if you detect something in your sample and it's not a contaminant, it means it came from the source of the specimen, but maybe perhaps it was not causing an infection. So a colonizer is essentially part of the flora, or if they didn't normally reside in the site as flora, but colonize it and it's not causing any problems. So that would be a colonizer. A good example is, some people may have Pseudomonas in the lungs, for example, and it may not, if they don't have any signs and symptoms of infection, it would be just a colonizer that normally would not be there in a healthy person. But, for example, someone with cystic fibrosis may have colonization of Pseudomonas in that region. Let's take a look at some terminology of human flora. So the first concept is microbiota. So microbiota refers to the collection of all living organisms present in a specific ecosystem or environment. So for example, the gut microbiota refers to all living organisms living in the human gut. And while dominated by bacteria, microbiota does include fungi, phages, archaea, and protists. And sometimes you may hear the term mycobiota, and that's specifically referring to the fungal component of microbiota, which is an emerging field in microbiota research. The next concept is microbiome. So this is essentially the combination of the microbiota as well as all the genes that come with these organisms. And because these genes, when translated into their product, can have an effect on the host, it is important. And to give you a perspective, the human genome has somewhere about 23,000 genes. So that's what's in our DNA. But the genes that come from the microbiome collectively is somewhere between 10 million genes to 200 million genes. So depending on an individual. And the last concept is the virome. So these were all living organisms, and since viruses are not living organisms, there could be viruses in the gut or as part of the flora, and not just viruses but also phages. And for most intact immune hosts, these will not cause an infection, so they will essentially be part of the flora, and they may have an effect on the health of the host. And that includes phages as well as viruses that infect other organisms such as archaea, but as well as viruses that could potentially infect the human cells when there is an immunocompromise. Now, it's important to understand the concept of prebiotics, probiotics, and postbiotics. So prebiotics comes before the probiotics. So prebiotics is essentially the food for probiotics. So this is the food for bacteria that are part of the flora. And for the most part, this is essentially fiber, but also could include other substances that are used by the bacteria. And probiotics, of course, is the live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. And probiotics could be part of food, but also could be available as pharmaceutical products. And last but not least, there is the concept of postbiotics, and this is essentially the byproducts made by these probiotics that confer the health benefit. So essentially, if we think that probiotics are good for you, the mechanism by which they are good for you could be pathways that are activated by some of these postbiotics. Now, in human flora, there are 5 phyla that essentially make up the flora. So these are in order of decreasing abundance. So the most common phylum is the Firmicutes or Firmicutes, some would say. The second one is Bacteroidetes. The third one is Proteobacteria. Fourth one is Actinobacteria. And in recent time, we also have Verrucomicrobia. And essentially, these are the 5 phyla. Now, you can see that when it comes to genus, there are many there are a lot of them in each of these phylum. The ones that are bolded are specifically the ones that are available as probiotics in the United States. So you will see that most probiotics products will include Lactobacillus species, and Lactobacillus has many different species that are not listed here. Occasionally, some products may include Streptococcus and Clostridium, so you just have to read the label to see if they include them. Currently, there aren't any probiotics products on the market that have Bacteroidetes. Some products have Bifidobacterium, and again, Bifidobacterium has many different species, so you will have to see if they're included. And in recent time, there is one product on the market that has Akkermansia available. When it comes to microbiota and probiotics, it's very tricky, so it's really hard to find for sure, which probiotic would be beneficial, if beneficial. And for the large part, it's because of individual differences in microbiota. So one person to the next, it's really different. The composition of microbiota is very different, and this is kind of part of why different individuals have different health outcomes, but it's it's really difficult to make sure that people have the same flora composition, and it may not even be necessary for everybody to have the same composition. But in general, we know that the more diverse the flora, the healthier the person would be.